Okay, so herpes, herpesviridae, multiplication cycle. All right, great. I'm recording. So this one is honestly pretty hard for me because after herpes, and after that, I never really studied too hard because I had other stuff going on, as in other classes. Stupid assignments that are all due on the 10th week and everything. Whatever, we'll push through. So remember, all multiplication cycles is the same thing: attach, penetrate, uncode, transcribe, translate, replicate, blah blah blah. But so with herpes, now herpes is just from the looks of it, it's a icosahedral virus that's enveloped. It has a double-stranded RNA genome, actually, or is it double-stranded RNA? No, it's double-stranded DNA. Okay, so that means that herpes is very similar to polyoma and papilloma viridae. Now, why Dr. Samuels didn't put this in the same let us learn this virus after the other two, I don't know why. I honestly think it would have helped us organize stuff better. But whatever, it is what it is. So this is a double-stranded DNA virus. Now here is a virion, obviously, and it's going to attach basically to cell receptors, and the cell receptors are these things called glucose. It's like glucoaminoglycosides or something like that, and they include like nectins and heparin sulfate receptors. And you can see here it's GD, GB, and GC receptors of the herpes virus. And when it attaches to there, then it can fuse and then uncoat. And here is the uncoated virion, which has a 150 kilobase double-strand DNA. So in order to get its double-strand DNA genome, obviously, if its genome's in the cytoplasm, it can't do much. It can't replicate. It definitely cannot replicate its genome because there's no DNA synthesis machinery in the cytoplasm. That's all in the nucleus. It can't make the mRNAs that it needs to make because all that stuff is in the nucleus of the cell. And this guy just simply does not. If I remember correctly, herpes does not encode its own polymerases, as far as I can tell. Or it does. We can see right here viral DNA polymerase. Okay, so viral DNA polymerase, whatever. Okay, so basically it injects its dsDNA into the nucleus so that it can be replicated and transcribed. So here's what makes herpes special. It's almost like it has three stages of transcription. So when we were learning polyoma and papillomaviridae, there was early and late transcription. When we were learning the viruses from rhabdovirus all the way to rhabdo, para, and orthomyxovirus, there was primary and secondary transcription. With herpes, there's immediate early, and late transcription. I like to shorten it to immediate transcription, early transcription, and late transcription. And basically, the earlier ones help facilitate the later ones. So you can see this is the genome. First thing that the herpes does is that it goes through immediate early transcription so that it can make the alpha mRNAs. So alpha is the first letter, so it gets transcribed first, and it makes these regulatory proteins, for example ICP4. It makes those, brings it back inside the nucleus so I can set the stage up for a next stage of transcription, and then it transcribes the beta mRNAs, the beta genes, and the beta genes have to go back outside. The beta mRNAs have to go back outside to get translated, and then these proteins go for nucleic acid metabolism. Basically, proteins that will set up the right conditions for DNA replication to occur. So you see viral DNA polymerase U, thymidine kinase, ICP, DNA, BP, all of these tell the cell to start replicating to metabolize or use nucleotides for viral DNA synthesis. So that goes back, those proteins go back inside. Finally, you can have DNA replication. So now you make a shit ton of progeny genome DNA. And this is where you have a temporal regulation of this. All these are pretty much temporal examples of temporal regulation. In order to transcribe and translate your gamma genes to do your late transcription, you need the early transcription to go first. In order to get the early transcription to go first, you need the immediate early transcription to go first. So you have all this progeny genome DNA, and then you have the late transcription, so gamma mRNAs. These code for the structural proteins, and the structural proteins get embedded, here. The structural proteins, some of them go to the nuclear pore, such as this. Others go to the cell membrane. It's basically prepping up the cell for export, for budding of the virion molecules. Because you can imagine, the DNA is too big to fit through the nucleus, right? It was able to fit through here because it had the help of virion molecule, but by itself, the DNA is helpless. It's trapped inside this nucleus. So what it does is, you have the gamma genes that are translated. They embed themselves at the membranes, at the nuclear pore. At the nuclear pore, inside the nucleus, you have a bunch of genome, a bunch of these structural proteins. Structural proteins plus genome equals a virus. And that virus goes from the nucleus into the ER. ER and a Golgi apparatus, they move it towards they export it to the cell membrane, and then they secrete out these virions. So herpes is able to be latent in its infection by residing in neurons, and it has these genes. It's not shown here, but it has these genes called latent associated transcripts, and these transcripts tell the herpes to slow down its transcription, to slow down its replication, slow down its translation. Basically tells its herpes virus to stop growing. You might wonder what the heck why would herpes want to do that? I thought it just wants to replicate and get out as fast as possible and destroy everything in its path. No, not necessarily, because if it does that, then the immune system would go into full swing, into full force, and kill off all herpes. So it uses these LATs, these latent associated transcripts, to purposely slow itself down so that it can hide within our cells. And then when it detects that we have a weakened immune system, from finals week, eating too much pizza, fries, and burgers, and going to sleep super late and waking up super early, coffee and everything, when we're feeling weak, now it can go replicate in full force. And our immune system is too weak to respond to it. So that's one thing to keep in mind. But in all, this is the multiplication cycle. Hope that helps. And on to the next video.